I'm Alice, and these are my friends.
Hi there, I am Alice, and together we build OBCI, that is short for Brain-Computer Interface. But wait, what does that actually mean for me?
Well, that's simple, Alice. That means I can talk to you only using my brain.
Oh, that means I am the computer?
That's right, and I'm the brain.
You said you can talk to me without speaking. I don't know how to read minds.
Well, you don't need to. We will be using SSVP.
I have no idea what you just said.
This is Alice. She is a humanoid robot that is mainly used in education and research. At TU, she had been sitting unused for quite a long time, so we decided to bring her back to life and use her in this project. In the next few minutes, we will show you how we made that happen.
Electroencephalogram data are time series recordings of the brain's electrical activity, like here in our example screenshots of waves for 4 different channels, which were measured non-invasively by electrodes placed on the scalp. Since these signals are in the microvolt range, they are very small and highly sensitive to noise. Raw EEG signals initially look chaotic and must be filtered and pre-processed before meaningful information can be extracted. Artifacts such as eye blinks, muscle activity, heartbeats, or even power line interference can easily distort the measurements. To ensure consistent electrode placement, we use the International 1020 system, which standardizes positions relative to underlying brain regions. Because SSVP responses originate in the visual cortex, we focused on occipital electrodes at the back of the head, highlighted in red. In this short clip, you can see the electrodes placed on these positions on my head, which were manually attached using the conductive adhesive paste to ensure stable contact.
Our goal is to take these raw brain signals and transform them into meaningful commands that can control a robot. To do this, we rely on a software layer called BrainFlow, which acts as the bridge between the EEG hardware and our own code. BrainFlow already takes care of most of the setup, including the initialization of the OpenBCI, the IoT connection, and recording the EEG signals. BrainFlow is a cross-platform library designed for biosignal processing. It supports many different devices, including OpenBCI, and provides a unified API. In our project, BrainFlow is the backbone that connects hardware to our BCI logic. Electrical signals are picked up by the electrodes. These signals are sent to the OpenBCI board, where they are amplified and digitized. The data is then transmitted wirelessly to the PC, where BrainFlow receives it and makes it available to our software in real time. But how do we use the information from our brain now to control Alice? For this, we use a phenomenon of the brain called SSVEP. SSVEP stands for Steady-State Visual Evoked Potential and describes the brain's response to specific visual stimuli. When a person observes a visual stimulus, such as flickering light from a screen, a response can be measured in the brain's electrical signals. The response occurs at the stimulus frequency and its harmonics. Once BrainFlow streams the data, it is passed to our BCI server. Inside the BCI server, raw EEG data arrives as a continuous time series. This data can either be live streamed or replayed from recorded files, which is useful for testing and offline experiments. To process EEG in real time, we define several timing parameters. To handle streaming data efficiently, we use a ring buffer. It stores the most recent EEG samples in a fixed size circular structure. This allows us to continuously update the analysis window without copying large amounts of data, like a memory that never gets full.
Before analysis, the EEG data is filtered. A bandpass filter keeps only the frequencies we are interested in and removes slow drift and high frequency noise. For the SSVEP detection, we use a 4 to 48 Hz bandpass filter. Most of the energy from blinks is in lower frequencies, so we use a different filter for blink detection. After filtering, the actual analysis of the EEG data can begin. To determine if the chunk contains a SSVEP signal, we use canonical correlation analysis, or CCA. CCA is a statistical method that measures the relationship between two sets of variables by finding linear combinations that are maximally correlated. In our case, we have X as the measured EEG data and Y as the reference signal at the frequency we want to detect. The CCA then computes the maximum possible correlation coefficient between linear combinations of those two datasets. During analysis, we compute the CCA correlation between the EEG chunk and each reference frequency we want to detect. We then compare the resulting correlation coefficients with thresholds. If one coefficient exceeds their predefined threshold, we assume that the person connected to the electrodes is currently looking at a stimulus flickering at that frequency. To confirm the detected signal and send an action to the robot, a blink detector is used. A blink or a nod produces a strong low-frequency signal in the EEG data. By computing the average power over the chunk, we can check if a confirmation gesture has been made. With this pipeline, we can reliably stream, clean, and analyze EEG data in real time.
The next part of our software architecture is the GUI. The GUI, short for Graphical User Interface, is connected with the BCI server via ZMQ. The connection goes both ways, with the most important data exchanged being the actual available frequencies and the detected frequencies, as well as the blink detection from the BCI server. The Python application is quite simple. The application first reads a configuration file in which different stimuli are defined with a text, a frequency, and optionally an action or a parent ID. The application will use this configuration to build a decision tree. Since the stimuli is vertical and horizontal then have a parent ID defined, they are interpreted as root nodes, being the uppermost stimuli. Other stimuli are placed accordingly below their set parent stimuli. Stimuli with a command variable set will also send out a ZMQ message with set variable. This stimuli tree is then linked to a GUI frontend written in CuteQML and will start displaying the root stimuli first. With the interface, it is now possible to navigate through the decision tree and confirm commands. Let's repeat the process one more time. A user connected with the OpenBCI will be staring at one of the presented stimuli. The recorded EEG will be sent to our BCI servers, which filters set signals and uses CCA to determine the most likely active stimuli. The result will be sent back to the GUI, which will target the frequency by painting it green. If now a blink is detected, the targeted stimuli will be selected and executed. If on top on the decision tree, it will now navigate to the second stage showing the next stimuli which had the corresponding parent stimuli. The GUI will notify the BCI server of the new presented frequencies for the CCA. If on bottom of the decision tree, it will now instead send a command out via ZMQ, which will be received by the last part of our software architecture, the NAO server. The NAO server is the component which transforms the command string sent by the GUI to actual movements on the robot. After initializing a connection to the NAO and to the ZMQ server, the main loop starts running indefinitely. Whenever the GUI server sends a confirmed command, the NAO server reads the message, translates it to its corresponding NAO command, and sends out the command after applying safety clamping. This command is being sent out via SSH on the IoT network of the NAO, which in response will execute any command it receives. The project is now complete.
Time for the fun part. How?